Mendel's law of segregation are the rules that Mendel figured out from his experiments about how it is that characteristics and different versions of characteristics are inherited throughout generations. Law of segregation says a sperm or egg carries only one allele for each inherited character because allele pairs are separated during the production of gametes. What are gametes? Gametes are basically sperm or egg. So what it's saying is that during sexual reproduction, when sexual reproduction is happening, obviously let's imagine this is male and this is female. During sexual reproduction, gametes, which are sperm or egg, are produced. And when sperm or egg are produced, even though parents have 2 copies of each gene, they only pass on one of them to the sperm or egg. So that's all the law of segregation means. And basically, segregation is synonymous with separation. So all it's saying is that when egg or sperm is produced, the homologous chromosomes that were in the parent cells, they are separated during sperm or egg production. So sperm or egg only has one of these alleles. So how do you produce the F1 generation? The sperm fertilizes the egg, and then you get the next generation. So then the next generation gets one allele from the father and one allele from the mother. So what is that? You must have heard that before. You may remember. It may brings out echoes of meiosis because we've seen that before. What is meiosis? Meiosis is a process through which egg or sperm are produced. So here we are. In this case, our example is a cell that has 2 pairs of homologous chromosomes. During meiosis 1, the homologous chromosomes line up at the center, and during anaphase 1, they are separated from one another. See, you're separating the homologous chromosomes. And then during the second division, you're going to get daughter cells after this, and you're going to separate the sister chromatids. But it is during meiosis 1 that you're separating the 2 copies of the gene. So really, even before knowing what chromosomes are, what mitosis or meiosis is, what genes are, Mendel, long before we learned about these things, he predicted this event. So the law of segregation correlates with the anaphase 1 stage of meiosis. Now, here's a little exercise for you to kind of better grasp these concepts. It's really not extra credit or something. It's just for you to learn, to maybe stop the video and complete the steps in this and the next steps. For this one, I want you to draw a pair of homologous chromosomes like I've drawn, and then figure out how many different types of gametes you get from each parent. How many different types of gametes you get from this one? How many different types of gametes do you get from this one? And what will the chromosomal composition would be? Now, if you take the gametes that are produced from this parent and that parent, you're going to get, that's the chromosomes for the offspring. I already drawn it. Okay? So now what do? You take this offspring and figure out how many types of gametes would you get from this offspring and draw it for yourself. Okay, so now let's take a look at the law of segregation in context of the crosses that Mendel performed. So here we have true breeding or pure breeding parental strain. So what does that mean? If it's pure breeding, that means that the individual is homozygous. Both alleles are the same. With a white plant, both alleles are the same. It's true breeding. Okay? So what happens during gamete formation? These two alleles are separated into gametes. How many different types of gametes does this parent produce? Only one. How many different types of gametes this parent produces? Only one. So then now, during the formation of the F1 generation, you bring the sperm and egg together, you get purple plants, and this purple plant has both a purple allele and a white allele. And then the next step was formation of the F2 generation. What has to happen for F2 generation to happen? Well, the plants, so this arrow means self-fertilization. Now this plant has both female parts and male parts. So you could say, if we just say this is the male part and this is the female part of the flower, they're both on the same flower. Okay? So this male part produces gametes. So it separates these alleles. The law of segregation separates them into separate gametes. And then the female, the same thing. It separates, because of the law of segregation, it separates these alleles into different gametes. So we got sperm and egg. And then we put these sperm and egg together. We get all sorts of different types of combination of these gametes, and it gives us the 3-to-1 ratio of purple flower to white flower. So now let's try to explain this ratio that we get. So here, if you look at the left-hand side, here's our cross again. What were the phenotypes we saw? We had white flowers and purple flowers, and then we crossed them together. The F1 generation was 100% purple, right? Why? Because the purple flower produces only one type of gamete. The white flower produces only one type of gamete. And then you put them together, you get only one type of progeny. That's why it's 100% purple, right? Okay. Now we're going to fertilize this plant by itself. And remember, we said the plant has both male parts and female parts. So each male or female part, the male part is going to produce sperm, and the female part is going to produce the egg. Okay? And there are two possibilities for sperm: purple or white allele. Purple or white allele. The same thing for egg. Purple or white allele. Now, in order to find out how we can get this ratio, we're going to produce something called a Punnett square. In a Punnett square, you put the gametes on the outside of this square. You produce a square and you put the gametes on the outside for the sperm and the egg, and you start combining them. So we get a purple from here, purple sperm, which is going to fertilize a purple egg, right? So we're going to get two purple alleles. So what is the color of this plant? Is purple. So we get 1 out of 4 purple. 1 out of 4 purple. I'm just going to write P. Okay? In here, we get a purple allele and a white allele. And what do we know? The purple allele is dominant over white. So the phenotype is going to be purple. Right? Okay. What about here? The same thing. We have 1 purple allele and one white allele. 1 out of 4 is going to be purple. In here, both of the alleles are white. So 1 out of 4 combinations is white. So I'm going to write W here because we're talking about phenotypes. Okay? So then 3 out of 4 are going to be purple. purple. 1 out of 4 are going to be white. 75%, 25%. That is called the phenotypic ratio, the ratio of the phenotypes of the individuals. Now let's look at what is the genotypic ratio. Okay, let's look at the genotypes in this case. So we have big P, big P, big P in the first one. We have big P, little p here, and a big P, little p here. So we have 2 of them. And then we have one here, little p, which is this one. Little p, little p. And these are ratios, they're not absolute numbers. So that's 1 out of 4. That's 2 out of 4. And that is 1 out of 4. Okay? I wanted to end this section by distinguishing between the phenotypic and genotypic ratio. So as you know, phenotype represents the expression of a particular genotype. So phenotype is what the organism looks like as a result of the alleles that it has inherited. So in this case, the phenotypic ratio is the ratio of the different phenotypes you have. You've expressed it here as 75% to 25%, but we can also express it as 3 to 1. So the phenotypic ratio for this cross, for a classical Mendelian monohybrid cross, is 3 to 1, purple to white. Genotypic ratio is the ratio of different genotypes that we have. So for the, again, F2 generation, let's take a look at different genotypes we have. We have big P, which is 2 purple alleles, and we have 1 out of 4. These are homozygous dominant. Okay, I'll make it longer so it shows that it's both uppercase. So this is big P. Then we have 2 heterozygotes. 2 out of 4 are heterozygote. Big P, little p. And then we have 1 homozygote recessive. So 1 out of 4 is little p. So therefore the genotypic ratio is 1 to 2 to 1. So in a monohybrid classic Mendelian cross, the phenotypic ratio is 3 to 1, and 3 is the dominant phenotype. And then the genotypic ratio is 1 to 2 to 1. One homozygous dominant, 1 homozygous recessive, and 2 heterozygote. It's really important that you understand the difference between a phenotypic ratio and a genotypic ratio.